I'd like to welcome everyone to the September 2020 Soils Network of Knowledge webinar. Now, what we're all here for is to listen to Dr. Corinne Celestina, and she's a research agronomist in the crop agronomy group at La Trobe University. She currently works on the GRDC National Phenology Initiative, and the aim of that initiative is to improve the accuracy of wheat and barley flowering time predictions. However, prior to this, Corinne worked with Southern Farming Systems in the high rainfall cropping zone, and in 2018 she completed her PhD at La Trobe University on understanding the drivers of the crop yield response to subsoil manuring, and from that came her paper, Attribution of Crop Yield Responses to Application of Organic Amendments, a Critical Review, which is where we've got today's presentation from. And this paper, I might add, earned Corinne the 2019 Soil Science of Australia Publication Medal. So we're very excited to have Corinne talk today.
So good morning, everyone, and I see some familiar names in the list, but for anyone that I haven't met, it's nice to sort of meet you over the computer and to have you here along to the webinar today. So as Abby introduced, this is a presentation that goes along with the paper that's linked in the handout section. Any other papers that I cite or link in my paper, if you're interested in, just contact me and I can send them to you as well. So to begin, organic amendments like manures, compost, plant residues, biochars, biosolids, they're often used in crop production systems as alternatives to inorganic fertilisers, to restore degraded soils, or to ameliorate soil physicochemical constraints. And we know that these organic amendments can indirectly affect crop yields via a really large range of non-nutrient effects. So they can increase soil organic matter content and soil carbon, improve soil biological activity, cation exchange capacity, structural integrity, and so on. But organic amendments can also provide large amounts of nutrients to the plant, which can directly affect crop yields via fertilisation effects. So yield responses to the application of organic amendments could be due to an organic matter effect. They could be due to the plant nutrients contained in the amendment, or to some combination of both of these factors. And it's important that we really separate these nutrient and non-nutrient effects so that we can identify where the yield responses are coming from. So here I've got two real-world examples of experiments that have been used to test the effects of organic amendments on crop yields. On the left, we have an untreated control and 3 experimental treatments with increasing rates of compost application. And the compost is being applied in an attempt to restore a degraded soil. And you can see that as the application rate increases, the yield increases as well. And on the right, we have a control treatment that's compared to deep ripping, and deep ripping with the addition of gypsum or poultry litter to ameliorate poorly structured subsoil. So the problem with these types of experiments is that their design is confounded. In the scientific sense, it's confounded, and you can't separate the effects on yield. On the left example, is yield increasing because the compost is having some sort of organic matter effect on the soil, and it's improving carbon and structure and biology? Or do higher rates of compost simply supply more nutrients to the crop? So with a linear rate of compost application, we see a linear increase in yield. In the example on the right, does the deep ripped poultry litter ameliorate that subsoil more than gypsum and ripping does? Or is the yield response because the poultry litter supplies more nutrients to the crop than the other treatments? It's important that we can properly attribute yield responses because we need to be able to advise growers about which management practices to use and where to spend their money. If they're chasing yield, should we be advising them to apply 15 tonne a hectare or deep rips and poultry litter, or can they achieve the same yield increase with an equivalent application of fertiliser? So without the right combination of treatments and the right assessments, it's easy to draw conclusions that might not be correct. For example, high rates of organic amendments applied to the soil, we know they can improve structure and root growth and water uptake. But it's also true that applying nitrogen to overcome a deficiency can also increase root growth and water uptake and lead to structural changes. So if you're just applying a treatment and measuring a response and not looking at the interaction and assessing it properly, it's hard to separate the nutrient and non-nutrient effects and attribute yield responses properly. The fact of the matter is that although soil organic matter and organic amendments are valued for their non-nutrient effects and the agricultural system services they provide, so all these things on the right-hand side of the scale there, buffering, disease suppression, cation exchange, water holding capacity, most of the direct impact on crop yields can be attributed to nutrients, whether that be the nutrient supply from organic amendments or nutrient turnover in the soil organic matter. And recently, there's been a number of reviews and meta-analyses that have highlighted the role of nutrients in the crop yield response to amendments. And I think they really challenged our understanding of the relationship between organic matter or organic amendments in crop productivity. So in the next few slides, I just wanted to show you what the global literature is telling us to highlight why it's so important that the experiments we do are designed and assessed properly. And you'll note my focus here, it's on crop yields. I'm not looking at experiments designed to improve the structure, designed to improve biology. I'm looking at the application of amendments with the aim of increasing yields. So the first review, it's a slightly older one from 2003, where Dorr et al. Reviewed the use of organic amendments in irrigated rice systems across Asia. And they showed evidence that yield trends in organic amendment treatments were indistinguishable from yield trends in NPK fertilizer treatments. But their key message was that many experiments that they reviewed weren't designed properly, and they didn't properly account for the macronutrient input from the amendment. So it was difficult to compare the advantages of inorganic versus organic fertilizers. So you can see in the two figures here that in the studies they reviewed, the grain yield of NPK fertilizer treatments on the x-axis was equivalent to grain yields in treatments with straw and manure added on the y-axis. So they're saying there that the yield trends, whether you're applying fertilizer or organic amendments, were equivalent. In a review of the Rothamsted long-term experiments, Ed Meads concluded that the use of manures may not necessarily enhance soil quality, which they define in terms of productivity or yield in the long term relative to applying the same amount of nutrient as fertilizer. And they present a version of this figure that shows that over 165 years of the broad bulk experiment, which is the longest running field trial in the world, that treatments with manure and inorganic NPK had the same long-term effects on crop production, so on grain yield. And this was despite the fact that the long-term manure treatments did lead to significantly higher organic matter, improved biology and better structure than the NPK fertilizer treatments. In a 2018 meta-analysis of long-term studies worldwide, Chen et al. From Massey University in New Zealand reported that there was no additional effect of organic amendments on crop yield beyond that of the provision of plant macronutrients. So in the top figure, you can see that when they pulled all the results of these trials, so 152 trials, and they analysed them together, they found that when organic amendments were compared to a nil control, there was on average an 81% increase in yield. But in the bottom figure, you can see that when organic amendments were compared to an inorganic fertilizer control, the mean yield increase was less than 1%. So in their meta-analysis, they also broke this down and they looked at things like the effective years of application, soil type, starting soil carbon, fertility, crop type. But the consistent message is that when organic amendments are compared to a control that supplies nutrients to the plant, the additional effect of the amendments over and above that nutrient effect is very small, generally less than 5 to 10%. A 2017 meta-analysis of European long-term trials found that organic inputs and or soil organic matter don't necessarily increase yields given sufficient nutrients are supplied by mineral fertilizers. And they reported that the additional yield effect of organic amendments over and above the effect of nutrients was found to be 1.4% on average. And in another review, the same author found that most of the studies that do reach a positive conclusion regarding the effect of increasing organic matter on crop yields don't control for the effect of macronutrients. So the figure on the left, you can see that the majority of studies that compared organic inputs to a fertiliser control, an NPK control, reported negative or no effects, as shown by the yellow and red slices on the pie chart. But when the organic inputs were compared to a nil control with no added fertiliser, the yield effect was always positive, as shown by the green shaded pie chart on the right. And finally, Schoening et al. Published a paper in Advances in Agronomy that really highlights the necessity of discriminating between nitrogen and non-nitrogen effects of organic matter and organic inputs. So they discuss how we need to change the way we think about the role of organic matter in crop production, and this concept can be extended to applications of organic amendments. So while, yes, it's true, organic amendments can improve soil properties, we can't underestimate the direct fertiliser effect of these amendments when they're applied to the soil. And as you've seen, there is actually quite strong evidence that nutrients play a much larger role than previously thought in the crop yield response to organic amendments, hence why we need explicit and clear differentiation between these factors that are driving yield. So together, what these reviews and meta-analyses are telling us is that in order to understand and tease apart the yield responses to amendments, you need to design and assess your experiments correctly. So we published a paper, and this is the one that's linked in the handout section, and this provides guidelines for organic amendment experiments that enables researchers to separate the nutrient and non-nutrient effects of organic amendments on a crop. These are identifying constraints to yield, the selection of proper controls, the use of appropriate sampling protocols, and the use of additional analyses to tease apart those yield responses. And we also discuss long-term experiments, controlled environment experiments, and simulation modelling, but I'm not going to cover those today. So the first guideline is recommended to anybody who's applying an organic amendment with the aim of ameliorating a soil physicochemical constraint or restoring a degraded soil. Before the amendment is applied, it's important to identify that there is a genuine and measurable constraint to crop yield at the field site. So this means identifying if plant growth is actually limited and to what extent. To do this, you need to do some soil sampling, obviously, to measure the soil properties of interest. So you might want to quantify the organic matter content, the pH down the profile, the sodicity of the subsoil. But you also need to carry out observations of crop rooting depth and soil water uptake to determine if these soil properties that you're concerned about are actually affecting plant growth. If there is a genuine constraint to crop yield, you should observe that root growth and water uptake is restricted or stopped. So it's not enough to say that sodicity is 15% or the pH is 8. You need to be able to demonstrate that these soil properties are negatively affecting plant growth before you go ahead and trial some intervention to ameliorate this constraint. So there's a number of methods to characterise root growth and water uptake. Soil scientists would typically use, say, a pressure plate apparatus. Agronomists would probably be more familiar with this method, which is outlined in the manual here by Burke and Dalgleish. Using this method, they measure soil water extraction by an otherwise healthy crop growing on your soil of interest, your constrained soil. So using this method, what you do is you measure a drained upper limit, which is the water content of the soil after deep drainage stops. And you do this using a watering system to saturate the profile and allow it to drain. And then you measure crop lower limit, which is the limit of what the crop can extract from the soil and you do this using a rain out shelter to isolate a section of plants and then measure soil water left over at maturity. And this will give you the plant available water capacity of a soil under a given crop. And that's what's shown in that diagram on the right-hand side there. So your plant available water capacity is that bucket of water that's bordered by the crop lower limit on the left in the red line and the drained upper limit on the right with the blue line. And compared to the lab method for generating a moisture curve with the pressure plates, using this method, you'll get a different profile for each crop grown on that soil because each crop type has a different rooting depth, they have a different ability to deal with the constraints and so on. So if you use this method, characterise your soil, what you'll see is that in an unconstrained soil where root growth and water uptake aren't limited, these lines for crop lower limit and drained upper limit for a given crop will meet at the maximum rooting depth of that crop. So if this diagram represents a wheat crop growing on a soil, you can see that our wheat roots go down to 2 metres and we're extracting that full bucket of water from the soil. On the other hand, if crop lower limit and drained upper limit meet at an unduly shallow depth, then you know that the plant hasn't extracted all the available water from the soil profile and that a severe constraint to root growth exists. And you might also see less severe constraints to root growth and soil water extraction, and that's seen when the crop's ability to extract water is substantially less than lab estimates of wilting point, but you don't see that cut off at a really shallow depth like you do with a severe constraint. So this method of soil characterisation, it's not easy to do. It is time consuming and can be difficult to set up. But it's one of the best ways to directly observe the effects of soil constraints on plant growth in situ. The second recommendation, and arguably the most important, is the selection of appropriate control treatments. So as I mentioned before, organic amendments can supply large amounts of nutrients to the crop. So in order to control for this enhanced nutrient supply, you should have a control treatment that supplies the equivalent rate of nutrients to the crop in the form of a synthetic fertilizer. In order to do this, what you need to do is you need to test your amendment to determine the total nutrient content because there's a huge variability in the nutrition supplied by different amendments. So if I use nitrogen as an example and some really rough calculations, an animal manure applied at 5 tonne per hectare might supply 100 to 250 kilos of N. Compost, maybe 30 to 150 kilos of N, depending on whether that compost is made from something like green waste or municipal waste versus a manure or a biosolid compost. And plant residues might range from something like 25 kilos of N from cereal stubble up to, say, 250 kilos of N in loosened pellets. So you can be applying significant amounts of nutrients in your organic amendments. So in this experimental design, you can have your nil control with no amendment, your organic amendment treatment, and then a synthetic fertilizer control. Now, because the amount and rate of nutrients released from the amendments can vary over time and obviously varies between different products, we recommend that with these synthetic fertilizer controls, you think about your application method. So use foliar applications, use slow release fertilizers, split your applications over time and even over years might be necessary to ensure that the nutrient supply matches the crop demand through the growing season and to avoid any toxicities and environmental effects that can come from applying higher rates of fertilizers up front. So if you recall our first example from the start of the presentation, we had a trial with the nil control and 3 increasing rates of compost application. So to properly control for the supply of nutrients in these 3 compost rates, you need to include a fertilizer control that supplies the equivalent total amount of nutrients as each of these composts, the 5, 10 and 15 tonne a hectare. Using these controls, you can then determine whether yield increase is due to the fertilizer contained in that compost or if the compost has some additional non-nutrient effect over and above the supply of nutrients. So if the yield of the compost and the matched fertilizer treatments is the same, which is what I'm showing on the left-hand side there, then that would indicate that the compost doesn't have any additional carbon, biological or possibly structural effect on the crop yield. So the yield responses are due to nutrient effects. But if the organic amendment is providing some benefit to the crop over and above that direct fertilizer effect of the nutrient, then the compost should increase crop yields more than just synthetic fertilizer alone. And that's what we're seeing in the right-hand panel there. So using that experimental design with the appropriate controls, you can tease apart those 2 effects on yield. In experiments, though, where the amendment isn't just applied to the soil surface, you're also incorporating it into the subsoil, you need to have additional control treatments to separate not only the nutrient effects of the amendment, but also the effects of placement and any interaction of amendment and placement. So in addition to the synthetic fertiliser control, you also need to include tillage control treatments, where the same rate of organic amendment and synthetic fertiliser is applied to both the soil surface and the subsoil. And this would give you a balanced factorial design with controls for tillage, nutrients, and placement effects. So in this design, we've got a full control and a tillage control. We have organic amendments applied to the soil surface and the subsoil, and we have our synthetic fertiliser control applied to the soil surface and subsoil. And if you recall our second example from the start of the presentation, we had that experiment with a control, a deep rip, and then deep ripping with the addition of gypsum and poultry litter. So to properly control for the nutrients supplied by the poultry litter, you should include a fertiliser treatment that supplies the equivalent amount of nutrients, but you also need to include for those tillage effects and any interactions. So the fertiliser and the poultry litter also need to be applied to the soil surface, and that gives you the design on the right-hand side there. So using these controls, again, you can tease apart what's driving the yield responses to the organic amendment. On the left-hand side, you can see if the surface-applied poultry litter yields the same as the deep rip poultry litter, then that would suggest that the placement of the amendment isn't necessary to increase yields, because you get the same yield increase whether it's on the surface or the subsoil. And if the fertiliser yields the same as the poultry litter, then that would suggest that yield increases are probably due to nutrients contained in the amendment, because you're not getting any additional yield effect from that poultry litter, from the organic amendment. Whereas on the right-hand side, if the deep placement of organic amendments is ameliorating some constraint in the subsoil, then there should be some positive interaction between the amendment and its placement in the subsoil. So that treatment increases crop yields over and above those achieved by surface broadcasting the same amendment or with synthetic fertiliser placed on the soil surface or in the subsoil. So again, without this, or without the inclusion of proper controls, it's really hard to tease apart whether your yield response is just due to nutrients, whether it's due to the organic amendment, or some combination of both of those factors. The third recommendation is to ensure that you're using appropriate sampling protocols to estimate or to measure crop yield. A common problem that we see is quadrite cuts or hand harvest cuts that are much too small or unconsciously biased and not necessarily representative of the crop response. So this is particularly a problem when organic amendments are incorporated into the soil profile, because as you can imagine, you end up with a spatially variable distribution of amendment and therefore nutrients, compared to a surface broadcast application where you're usually just evenly broadcasting that across the whole trial area. You can see in the photos here, on the left-hand side, the amendment is being ripped or injected into discrete bands. So we're having 2 bands of amendment and untreated soil in between. And in the photo on the right, you can see where, using this machine, the amendment is located in the soil and that the plant growth is pretty uneven as a result. So where the plant's growing on top of the amendment, it's bigger. Where there's no amendment, you can see the plants are smaller. So depending on the type of machinery, the width of the machinery, crops end up being sown over amended or unamended soil. So the sampling protocol that you use for yield has to take into account the spatial heterogeneity. So this figure, I'm just trying to show an example of an experiment where the amendment has been incorporated into the soil in discrete bands, and in this example they're 80 centimetres apart. And then the crop's been sown over the top, so that crop rows are either right next to the amended band or they're further away from it. And you can see that we've got some uneven growth in our plants there, because they're either sown on top of the amendment or over the unamended soil. So in order to properly capture this spatial heterogeneity, you need to harvest areas of the crop in sections that are as wide as the amended, so 80 centimetres wide, or in multiples of 80 centimetres. And in this example, this ensures that you get the correct ratio of amended and unamended rows sampled. So you don't over or underestimate yield. You get a proper approximation of yield. And regardless of where you place your quadrat or harvest your plants, you'll always end up with an unbiased selection. If your amendment was placed in bands 1.2 metres apart, you'd need to have sections 1.2 metres wide, and so on. So using this method, you avoid any issues that might come about with small hand harvest cuts or subsampling heads or anything like that. And the fourth recommendation is to carry out a range of additional measurements to help untangle these effects on yield. So we know that amendments can affect a range of soil and plant properties. So when we're investigating what's driving yield responses, you need to look at more than just grain yield per hectare to try and get the full picture. What exactly these measurements are will depend on the experimental question being asked. But they might include things like crop yield components and grain quality. This can tell you what's regulating yield responses in the plant. It's often very useful to calculate the nutrient balance by looking at the nutrients in the amendment, the soil and the plant at the start and end of the experiment. Plus, plant tissue nutrient concentrations can indicate whether there are any deficiencies that the amendment might be correcting, or in some cases, toxicities. You might want to look at crop water use or rooting depth if the amendment is expected to improve plant growth or soil structure. And usually you'll want to look at a range of soil properties if you're applying an amendment to improve soil conditions or ameliorate a constraint. So whether you want to measure bulk density, hydraulic conductivity, soil organic matter content, pH, something like that. So you need to select additional measurements in addition to your yield assessment to try and figure out what is driving the yield responses. So we've put together these guidelines so that we can properly attribute yield responses to organic amendments to nutrient or non-nutrient effects, or some combination of the two. And if you're not following these guidelines, particularly in regards to the selection of proper control treatments, you risk confounding your results. And if you're doing that, then you can be drawing incorrect conclusions regarding what's driving yield responses in your experiment. And then that might lead to incorrect recommendations for growers. So it's important we get it right from the start when we're designing and assessing our experiments. Oh, went backwards there, sorry. So just take-home messages. As we showed when we went through those reviews at the start, it's important that we do this because there is strong evidence that the yield response to organic amendments is often largely due to the nutrients contained in the amendment. And yes, although organic amendments do have benefits beyond their nutrient value, the contribution to yield is likely small. So we know organic amendments can improve yield potential, yield resilience. They affect the structure, the biology, they improve disease suppression, all these factors, but the contribution of those non-nutrient effects to yield is likely small. But unless you design and assess your experiments properly to allow the attribution of yield, you won't know what it is that's driving the response in your experiment. So with that, I'd just like to thank Latrobe Uni and GRDC who funded the work, and collaborators at Southern Farming Systems and Hartfield Site Group who also put in a lot of work for this paper and my PhD. Thank you.
Thank you very much, Corinne. That was fantastic and, very clear and concise. So, as I mentioned earlier, we have now time for questions. There is one to start off with. I think it might be more of a comment than a question, from Jason Condon. And Jason said, assumption that the treatment of fertiliser is different due to addition benefit over nutrients doesn't account for the mineralisation. It may be that you've underestimated the end release from the compost or organic matter source, so the difference could still be just a nutrient effect, not predicted by you at the start when you tried to match your fertiliser nutrient rate.
In terms of rates of mineralisation and rates of release, I think that's where, I'm not sure if this is what Jason was pointing out as well, but this is where the long-term experimentation comes into or becomes important, because these organic amendments, it's not a single season thing that you can apply and measure a response. We need to look at the balance of nutrient release over time and that long-term effect on the crop to fully understand what's going on when these amendments are applied. But Jason, if you wanted to add anything to that as well in the chat, go ahead.
I'm going to unmute you, Jason, because then you can speak directly to your question. There you go.
Oh, thanks for your talk, Corinne. That was great. Yeah, my point in my question was that difference that you have from the in your example compost versus the fertilizer treatments, which the way you presented it said that when you get a difference, the difference could be from the additional benefit of the compost, and it implied like a structural benefit or some other type of benefit. But from my experience, the hard bit is actually matching that fertilizer control to the organic matter amendment, because it's not, you can measure it, take the organic matter and measure the content of it, but it doesn't necessarily match what could be released, and so the difference could still be a nutrient difference, and that's the really hard bit of choosing those fertilizer controls.
Yes.
And just
okay. Yes.
what your opinion of that was, like, how do you get around that?
Yeah, I definitely agree, and it's easy for me to say, oh, match the nutrient content, but are you matching the we say total nutrient content, but you're not then matching the release over time. And we know the organic amendments, the nutrients there are usually not immediately plant available. So as you're saying, we have to wait for them to mineralize. So in that situation, if you're just measuring yield, you can't possibly get the full picture. And I think that's where looking at things like the entire nutrient budget and doing more soil sampling over time will help you try and complete that picture to see, is it a difference in, the mineralization of that compost, or is it a structural thing? Is it a biology thing? Is it something else? So if you simply, even just with the right treatments, if you're only measuring yield and you're only doing one season, you can still not understand what's going on there. So yeah, I agree wholeheartedly with what you're saying.
Yep, cool, thanks.
Further to that, Lucas's Van Sweeten, who's also a researcher with DPI, has sort of seconded that and has said the difference between organic amendments versus fertilizer could also be due to the priming of soil organic carbon, release of nitrogen from soil organic carbon. This is particularly relevant where N is in suboptimal rates. Rennick says, have you at any stage quantified the effect of inorganic nutrients on any increase in the bucket size of the soil?
Not me personally, Rennick, but I do have some papers in my collection and I can dig them out for you. So that's referring to my comment where I'm saying if you apply nitrogen to a deficient crop, an under-fertilized crop, you get bigger plants, more roots. The roots can affect the structure, which can potentially affect the bucket size, or the plant available water capacity of the soil.
There was a question about doing sort of soil profile characterizations before and after treatment. So are they something that you would advocate?
Yeah, so doing that profile characterization, what you're doing is you're assessing the bucket, the plant available water at that point in time. So if you're then going to apply a treatment to ameliorate the soil, ideally you'd want to then do that characterization at some stage in the future so that you can measure that, yes, we did increase the bucket and yes, we did positively affect soil structure and therefore plant growth and rooting capacity and the ability of that plant to take up water from the soil. But it is time consuming and it is hard to do, but in an ideal situation, yes, before and after would be good.
Okay, thanks for that. There is one more question. In the experiment for known nutritional effects, why compare poultry manure to fertilizers that only supply nutrients?
So the idea is that the poultry litter usually supplies a considerable amount of nutrients to the plant, but the poultry litter also has that whole range of other effects because it's an organic amendment. So if we can compare the poultry litter to a fertilizer control, then you're effectively any yield responses that the poultry litter generates over and above what you get with the fertilizer control, you can potentially attribute to non-nutrient effects. So the fertilizer control controls for the nutrients supplied by the amendment.
Thank you. Thanks for that question, Jose. All right, well, we might call it a day. Thank you very much, Corinne. I really appreciate that. So thank you, everyone, and we'll see you next month. Thank you, Corinne.
Thanks, Abby. My pleasure.